Welcome to Intro to Neuroanatomy. My name is Joanna Allardyce. I'm the lecturer in anatomy in the School of Allied Health. So during this session, we're going to think about the components of the nervous system. We're going to think about the general functions of the cerebral cortex, specifically or in more detail, looking at the primary motor and sensory areas. We'll then think about the meninges, the ventricles and cerebrospinal fluid circulation. Moving on to the blood supply to the brain and finishing up with motor and sensory pathways. Before I start, this website, neuroanatomy.ca, is an amazing and free online neuroanatomy resource. It has these interactive modules. They have everything from an introduction to very high level neuroanatomy. So you might want to have a look and you might find them useful to accompany your studies as you continue. So with that said, let's start off with the components of the nervous system. So I'm going to start off thinking about the cells. There are many different types of cells. We'll mention some of them in a little while. And I'm going to start by thinking about the neurons, as these are the cells that actually make things happen in the body. Right? So here we have an image of a typical neuron. So there are features that you'll find in most neurons. There are differences between different types of neurons, but these are the kind of typical structures that you will see on many neurons. Okay, so we have a cell body. This is usually quite large, obviously where all the processing is happening. So you'll find a nucleus within the cell body, and surrounding or extending from the cell body we have these little tree-like structures, the dendrites, and these will be making contact with other neurons, other neuronal cell types, receptors and other things depending on where they are in the body. We then have one long axon which projects from the cell body out into the periphery or out into another part of the brain. Perhaps it crosses over to the other side of the brain, and it's going to make contact via its axon terminals to synapse with, again, another neuron or a gland or a muscle fibre, whatever its function is. In many neurons, there are unmyelinated neurons, but many neurons will have a myelin sheath, and this myelin sheath is created by these little cells that wrap themselves around the axon. We call them Schwann cells. They're actually Schwann cells in the peripheral nervous system. In the central nervous system, we refer to them as oligodendrocytes, but they have the same function. They create myelin and they form this myelin sheath. What you might notice is each individual Schwann cell that's wrapped around the axon actually leaves a little tiny gap between each of the cells and between each of the myelin sheaths, if you will. And we call those the nodes of Ranvier, or Ranvier, however you want to pronounce it. So those are the typical structures that you would find on a neuron. There are also several kind of basic types of neuron that you might find, and some of these we'll be discussing when we talk about the sensory and motor pathways later, for example. Firstly, on the left hand side here we have a bipolar neuron. Bipolar neurons are often interneurons, so they are an intermediary neuron that might run between a sensory neuron and a motor neuron, or between two motor neurons. They tend to connect neurons together. So they have lots of dendrites, as we already mentioned, from a typical neuron, which then comes down into one dendrite, or you might actually call it the axon, but I would probably refer to it as the dendrite, which leads into the cell body containing the nucleus, and then out of the cell body we have the axon going down to the axon terminals. So that's a bipolar neuron or interneuron. We then have in the middle here a pseudo unipolar neuron. So essentially what it looks like is just one long axon with an offshoot for the cell body. These tend to be first order sensory neurons, so we find these in the periphery, and again we'll talk about these in a little bit more detail later on when we discuss the sensory pathways. But essentially, say you have a nerve coming from your big toe and you've just stubbed your big toe, and you feel the pain. The initial impulse that would have been picked up by one of these pseudo unipolar neurons and brought back to your spinal cord for further processing through the pathway. And then thirdly, you might recognize this because it's identical almost to the image I showed for a typical neuron. We call this a multipolar neuron, and these are very typical of motor neurons. So they have the cell body at one end with lots of dendrites coming in from many different places, and then they have the one axon that off it goes out into the periphery. So there are actually many cell types, as I did mention before, in the central nervous system, as well as the neurons themselves. We also have ependymal cells. These tend to be forming barriers. We have astrocytes, microglia, all these kind of supporting cells, if you will. And then, as I mentioned before, the myelin creating cells, the oligodendrocytes, which are essentially the central nervous system version of a Schwann cell. So then in the peripheral nervous system, we have the Schwann cells, and then we have also some satellite cells, which again are for support of neurons and other cells of the nervous system. So at this point, I'd love it if you pause the video and just have a go at labelling this cartoon image of a typical neuron. If you have a piece of paper, you can draw it. It's not particularly difficult, but have a go. So we've discussed the cellular components of the nervous system. So now let's turn our attention to the actual organs of the nervous system. So we're going to start off with the biggest and hopefully the most obvious part of the central nervous system, which is the brain itself. So here we can see three views of the brain. Over on the left here, we have a superior view. So imagine taking your scalp off and then looking straight down onto your brain from the top. That's the view we're seeing here. So what we see is the brain has two halves or two hemispheres that are separated longitudinally, i.e. from the front, which is here at the top of the image, to the back at the bottom of the image, by a longitudinal fissure. What you also might notice about the surface of the brain is it's very hilly. It's very, it's not smooth. There are lots of peaks and troughs, if you will. And we call each of these kind of lumpy parts, we call that a gyrus. And then the sort of gaps in between, we call sulci, or individual sulcus. The second image in the middle here, if we flipped over that first image and looked at the inferior surface or the undersurface or the base of the brain, this is what we would see. So there's a lot more going on in comparison to the superior view. So we can see, again, two hemispheres. There's that longitudinal fissure. We can also see a lobe that sort of projects down from the upper part. That's the temporal lobe. We'll talk about that in a second. You can also see the brain stem, lots and lots of blood vessels, some nerves, and then you can see the cerebellum as well, right at the back, or most posteriorly, to be anatomical about it. And then this third image is looking from a lateral view. So this would be the front here, and this would be the back. Okay. So again, we can only see one hemisphere here because we're looking from the side, but what you do get a nice view of again is the brainstem and the cerebellum. And again, we can kind of see, so this would be the superior part that we were looking at before. And then as we look from a lateral view, you can see that there's another long fissure here, or little slit, almost like a little cut. And we call that the lateral fissure. And that is actually separating this upper part of the brain from this lower part of the brain. This is a sagittal section of a human head. And what we are looking at is the cerebrum here within the brain case. Okay, so all of this that is highlighted in yellow is the cerebrum. So whenever I refer to cerebral cortex, I'm referring to the outermost shell, if you will, the outermost grey matter of all of this. So the cerebrum is split into 4 functional lobes. We have the frontal lobe, which is the biggest lobe, and as its name suggests, it is right at the front. And this runs all the way back to a little sulcus that we call the central sulcus. And the main functions that you'll find in your frontal lobe are motor functions. So your primary motor cortex is found within your frontal lobe. Also the motor portion of speech. And we also have this part here, the frontal association area. You may have heard of prefrontal cortex and things to do with kind of executive functions, decision making, motivation, kind of your personality and your behaviours tend to come from your frontal lobe. And then behind the frontal lobe we have the parietal lobe. This is a much more sensory lobe. So we have your primary sensory or somatosensory cortex, so the place that you actually perceive a sensation, whether it be pain or touch or a change in temperature. That's all processed by your somatosensory or primary sensory cortex. But also we have speech, so comprehension of speech and things like that, and also kind of sensory processing, so putting information together, bringing visual information, tactile information, auditory information, bringing that together to build up a picture of the world around you. And then right at the back, the most posterior part of our cerebrum is the occipital lobe, and the function of this lobe is vision, or things to do with vision. And then finally, that kind of lower lobe that I mentioned before, the temporal lobe, it sits just medial to your ears. So it makes sense that hearing would be a big function of this lobe. There's also other things like smell, and if we were to go deeper, there are things linked to memory and all sorts of things within the temporal lobe. But generally, hearing and things like that. Okay. So then if we were to take a cut through the cerebrum and look at the internal surface, these are four coronal sections through a brain. So you can kind of see the approximate place that that these slices have been taken from on the image on the left. So what you will be able to see is on the outer surface, you can see this kind of grey-brown stuff. This is the cerebral cortex that I was just talking about. And then you'll also see some paler, whiter matter, if you will. This is all made up of axons. So you'd find millions and millions of cell bodies within the outer cortex. The axons from those neurons would run through this white matter to get to wherever it is they're going. They might be going out into the periphery to your arms and legs, for example. They might be crossing over to the other side of the other cerebral hemisphere to tell the other side of your brain what's going on and things like that. But what you'll also see within the internal structure of the brain are some holes, which might seem strange, but they are supposed to be there. So they are our ventricles, and we'll talk about those in a little bit more detail shortly. And you'll also see some other kind of big lumps of grey matter deep within the cerebral hemispheres themselves. And those are referred to as either deep nuclei or basal ganglia. You've probably heard them called. So these are really, really important structures to do with initiation of movement and also cognition. Very, very, very important structures. Here we have the thalamus, for example. So your thalamus is like the sort of the gateway to your sensory system. So all sensory information passes through your thalamus. It's also really important in that process of initiation of movement. So we think of our cerebral cortex, or the primary motor cortex, as being responsible for making a movement happen. And that is physically true in that the neuron the pathway, should I say, that starts in your primary motor cortex is going to bring about a movement. It's actually going to bring about contraction of a muscle fibre. However, to initiate that process, you need lots of these deep nuclei to talk to one another, including the thalamus. So some very, very important structures going on deep within the cerebral hemispheres. So once again, if you are willing, why don't you pause the video and try to identify some structures and some features on this image. So if you can remember what the view is called, and how the brain is orientated, where is anterior, where is posterior, where is superior, where is inferior. If you can identify the lobes and you can actually do the same, if you will, for this image. So now we're going to discuss some specific regions of the cerebral cortex, or if you will, the functional localization of the cerebral cortex, namely the motor and sensory cortices. But before we do this, it's really important to understand that the human body is actually mapped out in a very distinct way in the primary and sensory motor cortices. So each area of the body has a very specific area within the cortex. So for example, the areas of your body that are more mobile or more sensitive, such as your fingers, for example, or lips, will occupy a much greater portion of the cerebral cortex than other areas of your body which move less or are less sensitive, say like your trunk, So what we end up with is a very, very strange looking pictorial representation of the human, where some of the features are far more pronounced than others. And we call this representation the homunculus, or the little man. So here we can see a coronal representation of the right hemisphere of the cerebrum, and drawn over it are the parts of the human body that are represented by each part of, or each area, of the cortex. What we actually see is, if this is the right cerebral hemisphere, then it would be the left half of the body that is represented on the cortex. And actually, what you also might notice is that the body is upside down. So at the superior aspect of the cerebrum, and dangling down into the longitudinal fissure, we see the feet and the legs. And then as we move down and more lateral, away from that longitudinal fissure, we'd see the trunk, we'd see the hands and the fingers. And remember how I said we do a lot of fine movements with our hands and with our fingers, so they take up a relatively huge part of the cortex because of that. So that just essentially means that there are many, many, many more neurons that are specific to our hands than there would be to, say, our chest or our trunk as a whole. And then we see our face here. So again, a humongous mouth, because we move our mouth a lot, whether it be for speaking or eating, and all of those things. And then we can see just beneath our chin area, there's like the larynx, the tongue, and the upper esophagus, things like that. So that's the primary motor cortex. And actually the primary motor cortex is found in this little strip here. This is the precentral gyrus, or the primary motor cortex, or the primary somatomotor cortex. There are lots of different names. It doesn't matter what you choose, just choose whichever one you can remember and go with it. Whether you call it the primary motor cortex, which is what I tend to call it, but what you might see in textbooks is it's referred to by its location, which would be the precentral gyrus. Pre meaning before, central referring to the central sulcus that is running just posteriorly to it here. So the precentral gyrus, primary motor cortex, or like I say, you might see somatomotor, which essentially just means moving the body. And then we can do the same for the primary sensory cortex. So once again, we have that little coronal section of a right cerebral hemisphere on the cartoon here. And the body, so with the left hand side of the body that is represented on this hemisphere and in the same orientation. So the feet would be dangling down in the longitudinal fissure, actually along with the genitals. There you are, right under the toes there. And then with the kind of head and neck structures further towards the lateral aspect and the lateral fissure of the cerebrum. So again, we have sensitive fingers, very sensitive to touch. Therefore, our fingers and our hands in general would take up a much larger part of the cortex, the sensory cortex, as well as parts of our face as well. So if we think about the sensitivity of your lips, they take up a relatively huge area of the cortex compared to other structures which are much less sensitive, say like your forearm. Now, so going back to this lateral view here, we can see that the primary sensory cortex would be this strip of cortex here. So it lies in the post-central gyrus, post meaning behind, again that central sulcus that runs down here. So that's where we find it. We find the primary sensory cortex running directly behind the primary motor cortex. So the next structure, moving on from the cerebrum now, the next structure that we would be interested in for the central nervous system is the cerebellum. So we can see the cerebellum on this sagittal head section, just nestled in underneath the cerebrum, right at the back, kind of just above the neck, if you will. Excuse me. So the cerebellum, again, like the cerebrum, has two hemispheres. They talk to one another. They also communicate with the cerebrum and also via the spinal cord with the rest of the body. So the cerebellum is really, really important when it comes to control of movements and particularly muscle tone and posture. And it also helps with kind of thinking about, if you were, say, throwing a ball, things like trajectory, power, speed. So when you have issues with the cerebellum, it tends to affect your movement. So you might see someone with an ataxic walk or a drunkard's walk, it tends to be referred to, where people kind of walk with a sort of shuffle and a very wide stance, and they have that kind of drunk appearance about them because it's so important in controlling movement. And then the other part of the central nervous system, very important part, 3 parts actually, is the brainstem. So that's shown in green here, sitting directly anteriorly to the cerebellum and inferiorly to the cerebrum. And as I say, it's in 3 parts. So the upper part here, that is in direct contact and direct communication with the cerebrum, that's the midbrain. Beneath that we have this bulging part of the brainstem, which is the pons. Pons meaning bridge, and it essentially bridges between the 2 hemispheres of the cerebellum, so very, very closely linked with the cerebellum. And then beneath that we have this kind of cone-shaped part of the brainstem, which is the medulla, or the medulla oblongata, Either is fine. So it has this kind of funnel shape as it kind of decreases in size to become the spinal cord, which we can see running down the vertebral column there. And then, so thinking about the spinal cord itself, it runs all the way from the medulla through the vertebral foramen, or the vertebral canal of the spine, or the vertebral column, and it actually ends, so the spinal cord itself ends at around the vertebral level of L2, the second lumbar vertebra. Ends in this little cone shape. We can see it better on this dissection, so you can see how the spinal cord is quite thick as it runs down, and then it starts to taper and taper and taper to a very fine little structure that anchors it to the end of the vertebral column. That's the phylum terminale if you're interested. The reason it does that actually is all to do with how we develop. So your spinal cord, when you were a fetus in your mother's uterus, was already the length it was always going to be. So at one point the spinal cord fit the entire vertebral column, but then as you grew, your vertebral column got longer, but your spinal cord doesn't get any longer, which is why there is this discrepancy between the level of your spinal cord and the length of the spinal cord and the length of your vertebral column. It is also the reason why we can do things like epidurals and stuff like that, because you can introduce anaesthetic below the level of the spinal cord and just anaesthetize the nerves that run below, i.e. to the pelvis and to the legs. So we've covered the components of the central nervous system, and now we're briefly going to consider the peripheral nervous system. So any nerve that you have come across, any named nerve that you've come across during your studies up to this point, say the median nerve when you're thinking about carpal tunnel, or the sciatic nerve of the lower limb and the problems that can arise through entrapment of that, or even the phrenic nerve controlling the diaphragm. These are all examples of spinal nerves. Usually several spinal nerves have joined together to form these named nerves. And they're all part of the peripheral nervous system. So that essentially means that they connect to the nervous system. Of course, they have to get back to the central nervous system somehow. For us to be able to make a movement or be aware of a sensation, our brain has to be involved. But the actual neurons themselves are in the periphery. They're only attached to the central nervous system. So because these nerves are arising or going back to the spinal cord, we refer to them as spinal nerves. So as I say, any of those named nerves that you've come across will be spinal nerves. There are also nerves that arise from your brainstem or the actual forebrain itself of the cerebrum, and we call these cranial nerves. They're very similar to spinal nerves, and again you'll have come across cranial nerves thinking about the facial nerve and Bell's palsy, thinking about laryngeal function perhaps, and things to do with vagus nerve, stuff like that. So we call those the cranial nerves. And we'll talk about the origins of those in a minute. It's also important to note at this section that part of your peripheral nervous system includes your autonomic nervous system. So we have two strands of your autonomic nervous system, which are essentially controlling functions that you are not aware of, things like digestion, your heart rate, how much you breathe, how much secretion comes from your salivary glands, stuff like that. You're not in conscious control of that, which is why we call it autonomic. So there are two strands, as we say, the parasympathetic and the sympathetic. Now, neurons from these two systems run with spinal nerves, they run with cranial nerves, or they can even hitch a ride on blood vessels to actually get to their target. So the parasympathetic system arises via 4 cranial nerves that we'll talk about in a little minute, and also the spinal cord segments S2 to S4. And then the sympathetic system arises from spinal segments T1 to L2 exclusively. So this tends to be the case in everybody. It doesn't tend to deviate. So, thinking about the cranial nerves themselves, there are a few questions that we ask ourselves when we're discussing the cranial nerves in an anatomical context. So we want to know how many are there. So there are 12 pairs, 12 on the right, 12 on the left. What are their names? So that I'll give you an overview in a little minute. What are their functions? Whereabouts would we find them on the central nervous system? Are they from the medulla? Are they on the midbrain, etc.? And we also tend to think about the hole in the skull, or the foramen in the skull, that they traverse in order to get to or from the central nervous system. So why don't you pause the video for a minute and see how many cranial nerves you can remember. Can you remember what their functions are? And then there'll be a review slide afterwards. So this is the overview of cranial nerves. We have 12 pairs controlling either movement or sensation within the head and the neck. Also, as we mentioned before, there are 4 of these cranial nerves which are highlighted that have parasympathetic functions on top of either motor, sensory, or both. So there's the oculomotor nerve, cranial nerve 3. So this is responsible for pupil constriction. In bright light, your pupils constrict. That's your oculomotor nerve, the parasympathetic part of your oculomotor nerve kicking in there. The facial nerve, so that controls some of the salivary glands, and also your lacrimal gland, which creates tears. So it functions in salivation and lacrimation. The glossopharyngeal nerve, that is also involved in salivation. Actually, a salivary gland that lies on your cheek, the parotid gland, a huge salivary gland. And then finally the vagus nerve, cranial nerve 10. This is very, very far reaching. So it has some motor and sensory functions within the head and neck itself. Very important to do with your larynx, your pharynx. But it also, the parasympathetic part of the vagus nerve also runs to your heart and decreases heart rate, it decreases respiratory rate, and also increases digestion. So I mentioned the cranial nerves tend to function within the head and neck. Well, the vagus nerve would be the exception to this rule, and it's only the parasympathetic portion that actually leaves the head and neck and travels down through the thorax, controlling heart and lungs, and into your abdomen, controlling your gut. And then here we have a representation of on the left-hand side the position of each of the cranial nerves on this cartoon image of the brain, and then on the right-hand side the foramina in the skull that the cranial nerves traverse. So let's move on to look at the meninges now. So these have very important protective and some physiological roles within the central nervous system. There are 3 layers of meninges. The outermost layer is the dura mater, or just the dura is fine. This is like a very, very thick, fibrous, very, very strong sac that surrounds the brain and the spinal cord. Within the brain itself, it has 2 distinct folds. One in the longitudinal fissure that we can see here. This is the falx cerebri. And then there's another fold that separates the occipital lobe above from where the cerebellum would be sat below, and it's this structure here, and it's called the tentorium cerebelli. And within these folds there are really huge veins which we call the dural venous sinuses. So they're draining venous blood from the brain and returning it back to the systemic venous system. So you might have come across extradural hematomas or subdural hematomas, so bleeds within the cranium. So these are not bleeds on the brain as such, they are bleeds from these dural venous sinuses. That's not to say that they can't cause catastrophic problems, but they're not vessels that are supplying parts of the cortex, so parts of the brain aren't immediately affected by these types of hematoma. Now if we look at the next layer, this is the arachnoid mater, or just the arachnoid layer, and we can see it here surrounding all of the cerebrum. You can see it on the surface of the cerebellum and also on the brainstem. And essentially running beneath this layer are all the blood vessels that are actually supplying the brain itself. So you'll have arteries that we'll discuss shortly running in the subarachnoid layer that then actually supply the cortex of the brain itself. So again, you might have heard of subarachnoid hemorrhages. So this would be a bleed in one of the arteries or blood vessels within the subarachnoid space, and obviously this can have catastrophic effects on function, on brain function. Also within this kind of space that lies beneath the arachnoid layer, we call it the subarachnoid space, we have CSF. So cerebrospinal fluid is floating within this layer, essentially bathing the skull in this fluid. And there are connections between the arachnoid layer and the subarachnoid space where this CSF sits with the dural venous sinuses. So this is a way of getting the CSF back into the venous system so it can be recycled, as CSF is constantly being produced. And then the innermost layer is the pia. This is very thin. It means delicate mother. If you if you want to be specific. And the pia is actually completely adhered to the surface of the brain. So on this side of the brain here, we can see the right side of the brain, we can see the arachnoid layer still intact. And then on the left hemisphere of the brain here, the arachnoid layer has been removed, and we're now looking at the pia. But you can't really tell the difference between the pia and the cortex of the brain itself with the naked eye, because the pia follows the sulci and the gyri so very closely. So earlier on we mentioned that there were some holes in the brain. If you remember back, we saw a coronal section of the brain when we were looking at deeper structures, and I mentioned these holes. So these are the ventricles. Essentially the brain and the spinal cord develop from a tube. So there is a hole within this tube, so as the brain gets bigger and folds in on itself, essentially these holes turn into what we know as ventricles. These ventricles are really important, again, for protection of the brain. They are filled with cerebrospinal fluid, so they allow the brain to be buoyant, and they protect it from being bashed about within the brain case itself. So this CSF is actually circulating continuously throughout the ventricles and communicates with the subarachnoid space. So CSF is created within the ventricles, which we have, and we can see here. This is the lateral ventricle. This is the third ventricle, and they communicate through this little hole that you can just see a tiny bit of here. That's the foramen of Monro. We then see another teeny tiny channel. This is the cerebral aqueduct, and the third ventricle communicates with the fourth ventricle here, which then continues down the spinal cord as the spinal canal. Also in this region of the fourth ventricle, there are some little holes known as the foramina of Luschka and the foramen of Magendie. And these are little apertures where CSF can leave the fourth ventricle and actually get out into the subarachnoid space and then surround the outer cortex of the brain, brainstem, cerebellum and spinal cord. So the whole of the CSF, central nervous system, sorry. So, like I say, the CSF is continuously being circulated. So it's created within the ventricles, particularly the lateral ventricle, and it's created by these tiny little plexus of capillary-like structures that we call the choroid plexus. And then, so it's created there, and then it passes through the ventricular system out to the subarachnoid space, where it then is sent back to the venous system via these little out pockets, and you can see them on the cartoon here, these little out pockets of arachnoid mater that project into the dural venous sinuses, and we call them arachnoid granulations. And from here the CSF is able to be released back into the venous system, and the system just carries on circulating and circulating. So now we're going to move on and think about the blood supply to the brain. It's obviously hugely important for the brain normal function and also from the point of view of stroke and things like that. So, first things first, where does the blood supply to the brain originate? So, all the blood that enters into the cranial cavity arises from the base of the neck, or even from the arch of the aorta itself. So there's a long way for this blood to travel. So we either get blood from the common carotid arteries, which you can see the left common carotid here, and you can see the right common carotid here, or through a branching of the subclavian artery, which is here, going to the upper limbs, and you can see the branch here, the left vertebral artery and the right vertebral artery. So there are actually two routes by which blood gets to your brain. The first route we call the carotid system, and as you might be aware, the blood travels via the common carotid arteries. It runs up the side of the neck, and then we see a bifurcation of the common carotid artery into a smaller external carotid artery, which is going to supply your face, your scalp, the dura, also structures within your head, but not your brain. And then the internal carotid artery, which is going to continue up. It's going to enter your cranial cavity through the carotid canal and will supply the brain itself. The second route by which the brain is supplied comes from that second branch that I mentioned, that comes from the subclavian artery. Here it is on the left, and that's our vertebral artery. So there's the left vertebral artery and there's the right vertebral artery, and they travel up the neck, but this time they actually travel within the transverse foramina in the cervical vertebrae. They enter into the cranial cavity via the foramen magnum, and then the two systems, the internal carotid system and the vertebral system, are both going to unite, and they form this little circuit on the base of the brain called the circle of Willis. So when the vertebral arteries enter the cranial cavity, they converge on each other and they form one artery, that's the basilar artery. So that vertebral system can also be called the vertebro-basilar system. And it's branches of that basilar artery, that union between the two vertebral arteries, that give off branches that will connect with branches of the internal carotid to form the circle of Willis. So let's look at that in a little bit more detail here. So here are the cut ends of the internal carotid artery, and here are the vertebral arteries, 1, 2, converging to form the basilar artery here. So what we see is a posterior cerebral artery going to the left and a posterior cerebral artery going to the right, and then we can see some posterior communicating arteries. And it's these posterior communicating arteries that actually unite with the internal carotid artery to unite the vertebro-basilar and the internal carotid system. We then form a complete loop of the circle of Willis via this artery here, the anterior cerebral artery, which that's the left anterior cerebral, and here's the right anterior cerebral, and then the anterior communicating artery that sits between those two anterior cerebral arteries. So that's the circle of Willis. We have the internal carotid artery, the anterior communicating artery and the anterior cerebral arteries, The middle cerebral artery, which we're going to talk about in a minute, that's essentially the continuation of the internal carotid artery. The vertebral arteries, the basilar artery, and the posterior cerebral arteries and posterior communicating arteries here. So, the blood supply to the cerebral cortex is extremely important, and it's supplied by three main arteries. We've already mentioned them on the previous slide. And those three arteries supply three distinct territories on the cerebral cortex. So firstly, we have the anterior cerebral artery, and if we look at a lateral view from the brain, so this is a lateral view of the left cerebral hemisphere, anterior over here, posterior over here, superior and inferior. So the anterior cerebral artery supplies this relatively thin strip of cortex on the upper lateral or the superior lateral aspect of the brain. And the anterior cerebral artery is a branch of the internal carotid. We then have the middle cerebral artery, which supplies a relatively huge proportion of cerebral cortex on this lateral view. So lots of the frontal lobe is supplied by middle cerebral artery, lots of the parietal lobe is supplied by middle cerebral artery, and the whole upper half of the temporal lobe is also supplied by middle cerebral artery. And as I said on the previous slide, the middle cerebral artery is essentially a continuation of the internal carotid artery. So it comes from the carotid system. The third artery is the posterior cerebral artery, and this supplies everything else that I haven't mentioned on the cerebral cortex. So it supplies the a small portion of the parietal lobe, the occipital lobe, and that whole inferior half of the temporal lobe. And it's the posterior cerebral artery that comes from the vertebro-basilar system. So this pattern isn't unique, it's typical between us all. So this pattern is typical from human to human. Now the exact demarcation lines between anterior cerebral and middle cerebral, etc., may be slightly different from person to person, but the general pattern tends to be the same. Again, there can be exceptions to the rule. There's always exceptions to every rule, but for the most part, that would be true. So we saw the territories of the anterior, middle and posterior cerebral arteries on a lateral view. So then we'll have a look on a medial view here. This is a medial view of a right cerebral hemisphere. So here is, this would be anterior, this would be posterior, superior and inferior down here. So now what we see is the medial view of the cerebral cortex. So this is everything that would be within that longitudinal fissure that we mentioned earlier on. We can also see the half of the brainstem and one of the right cerebellar hemisphere as well. You can also see the ventricles that we were referring to earlier. So from the point of view of supply of the cerebral cortex here, the anterior cerebral artery now supplies the vast majority of the cortex within this view. So it supplies all of that frontal lobe on the medial side, all the way back to supply the majority of the medial part of the parietal lobe. The middle cerebral artery, on the other hand, on the previous view supplied a lot of cerebral cortex, but actually on this view it only supplies this very anterior part of the temporal lobe. That's the temporal pole. And then the rest is supplied by the posterior cerebral artery. So again, all that medial aspect of the occipital lobe, a little bit of parietal lobe, and the rest of the temporal lobe. So those are the territories of the 3 cerebral arteries. And it's important that we know this, and so then if we think about the 3 cerebral arteries, they will then further divide into smaller branches as they travel deep within the structure of the brain and supply some of those deep nuclei or basal ganglia and other structures that we mentioned earlier, the internal capsule, and all those really important structures within the brain. And that is summed up on this slide here. So here we have a coronal section showing branches of the anterior cerebral artery in blue, branches of the middle cerebral artery in this more yellow colour, and branches of the posterior cerebral artery in red. And then we have this other artery that is important and certainly worth mentioning, which is the anterior choroidal artery. And this is a direct branch from the internal carotid artery and is supplying an area deep within the brain. The brainstem and the cerebellum are also supplied by branches of the circle of Willis, more specifically the vertebrobacillar part of the circle of Willis. So we can see 3 main arteries that supply the cerebellum. This is the superior cerebellar artery, supplying, as its name suggests, the superior portion of the cerebellum. Then we have the anterior inferior cerebellar artery and the posterior inferior cerebellar artery, which are both supplying the inferior part of the cerebellum, either anteriorly or posteriorly. The brainstem itself is then supplied by little bacillar arteries, pontine arteries that arise from the bacilla. For the final part of this lecture, we're going to discuss motor and sensory pathways. So essentially, the pathways bring everything together that we've discussed. They bring together the central nervous system with the peripheral nervous system and link everything up. So this is a cross section of the spinal cord, and the areas that represent different motor and sensory pathways are illustrated in either red for motor or descending pathways, and blue for sensory or ascending pathways. You can call them whichever you fancy. So as you can see, there are a lot of them, but I don't want you kind of to panic about that. We're only actually going to talk about the voluntary pathways today. The other pathways we don't tend to go into a lot of detail about. It's important to know they exist and have an idea of what they do. But for the remit of this lecture, we're just going to talk about the voluntary motor and voluntary sensory pathways. So with any tract, any of the tracks we're going to talk about today, whether motor or sensory, there's a few questions that I would ask yourself. to help you kind of keep you on the right track, if you will. So how many neurons are in the pathway? If it's a descending pathway or a motor pathway, there's going to be 2 neurons. If it's a sensory pathway, there are going to be 3. So how many synapses would that mean? 1 in terms of motor pathways and 2 in terms of sensory. It's also really important to remember that the right hemisphere of the brain controls the left side of the body, and the left hemisphere of the brain controls the right side of the body. So at some point, the neurons within these pathways, whether they be motor or sensory, must cross the midline to get to the other side. So we have to think about where this crossing or decussation happens, and also where the neurons are synapsing. So first of all, I'm going to discuss the motor pathways. So there are 2 main voluntary motor pathways. We have the lateral corticospinal tract, which is shown in this cartoon on the left, and we have the anterior corticospinal tract, which is shown on the right here. So if we want to move something, what might we want to move? A finger. You might want to move a toe. I don't know. Let's move something in the hand. Okay. So when we want to move our hand, for example, the impulse is going to start within the primary motor cortex of the cerebrum. Okay. So if you remember that strip of cortex that we talked about earlier, which was the precentral cortex or the primary motor cortex, the impulse is going to start there. Okay. It's then going to run down through the axon of an upper motor neuron. So that's the first neuron in the pathway. Okay. So the impulse is going to run down the axon through this area of white matter that we call the corona radiata, and then it's going to pass through the posterior limb of the internal capsule, and it's going to pass into the brainstem via the cerebral peduncle, which essentially the cerebral peduncle is just the connection between your cerebrum and your brainstem, because they're connected via lots and lots of axons. So once in the midbrain, the axon for our lateral corticospinal tract fibre runs in the anteriormost part of the midbrain. We call it the crus cerebri. So it's going to run through the crus cerebri of the midbrain, then it's going to run through the anterior part of the pons, and then the anteriormost part of the medulla. And in the medulla, we call the areas where these lateral corticospinal tract fibres are running the pyramids. So you might also have heard this pathway referred to as the pyramidal system, because the axons are running through the pyramids of the medulla. So what's important is in the lateral corticospinal tract, at the position of the pyramids in the medulla, that is where we get our decussation of our upper motor neuron. So the upper motor neuron crosses to the opposite side of the medulla and then continues its descent down the spinal cord on the opposite or the contralateral side. Okay, so our signal in this pathway started on the right side, on the right cerebral hemisphere it stayed on the right side of the central nervous system until it reached the pyramids within the medulla, and then it crossed over to the left side of the body, or the left side of the medulla, to control the left side of the body eventually. So now it carries on down the spinal cord in what we call the lateral corticospinal tract, and it runs down to the particular level that the spinal nerve that is going to control your, we said hand movement, didn't we? So we want to get down to around T1 level. Okay, so the neurons that are going to come out of T1 level and that are going to run in your median nerve and in your ulnar nerve, etc., are going to come from around the T1 level for movements of your fingers, say. So the neuron passes or descends down the spinal cord all the way to the level of, let's say, T1, and at that point the neuron exits from the lateral corticospinal tract and enters into the grey matter of the spinal cord. Once it's in the grey matter of the spinal cord, in the ventral horn of the spinal cord, we see our synapse between the upper motor neuron and the lower motor neuron. And from there, the lower motor neuron, which is a peripheral nervous system neuron, passes out via the ventral root and enters into the T1 spinal nerve. It'll then join together with spinal nerves from C5 to T1 and form your median nerve. Run all the way down your upper limb to whichever muscle we're controlling our fingers with in the periphery. So that is the lateral corticospinal tract. So how many neurons were in the pathway? There are two, an upper motor neuron and a lower motor neuron. How many synapses? There are one. The synapse happens at the level at which the particular spinal nerve that is going to control the muscle or the movement occurs. Where do the neurons cross? They cross at the level of the pyramids within the medulla. And the location of the synapse is in the ventral horn of the spinal cord, at the level at which the spinal nerve is exiting the spinal cord. So that's the lateral corticospinal tract. Then the anterior corticospinal tract is very, very similar, almost identical, but there is one main difference, and that is that the decussation of the upper motor neuron in the anterior corticospinal tract decussates not at the level of the pyramids, but actually at the level of the spinal nerve at which the synapse with the lower motor neuron is going to occur. That is the only difference. So if we do the same, we're going to move a hand, we're going to go down to T1 level. So our impulse starts in the, if we remember our homunculus as well, in the hand area of the cerebral cortex on the right side of the body, because we're going to move our left hand. The impulse starts, it runs down the axon, through the corona radiata, through the posterior limb of the internal capsule, through the crus cerebri of the midbrain, through the pons, straight through the ipsilateral pyramid, stays ipsilateral, and this time, instead of running in the lateral corticospinal tract, it runs more anteriorly in the spinal cord on the ipsilateral side in what we call the anterior corticospinal tract, all the way down to the spinal level of T1, where it then crosses over via the anterior white commissure, enters into the left ventral horn of Gray in the spinal cord, synapses with the lower motor neuron at the level of T1, and then that T1 spinal nerve goes off down the arm, and you move your left hand. So the only difference between the lateral and the anterior corticospinal tract is the position of the decussation. The vast majority, 75 to 90% of neurons, of descending neurons in this voluntary motor pathway run in the lateral corticospinal tract. It is a much, much bigger area. You can see it here. And then the remaining 10 to 25% run in the anterior corticospinal tract. So that's the motor. And actually, it is important to remind yourselves when you're thinking about these tracts of myotomes and dermatomes. So a myotome refers to a group of muscles that are innervated by nerves of nerves that come from a single spinal root. So for example, I was talking about hand movement then, wasn't I? Moving your fingers here, so opening and closing your hand, for example, that is brought about by T1. So the myotome for finger movement in that case would be T1. A dermatome on the other hand refers to an area of skin, so like a strip of skin that is innervated by nerves that arise from a single spinal root. So essentially one spinal nerve. And what we end up with the dermatome map is more of a stripy appearance. It's lovely over the trunk. So each spinal nerve corresponds to a little strip of skin. Whereas on the arms and legs it's a little bit more complicated, but that is the little map. And so just something to have in your mind. And then we'll think about the sensory tracts. So these are the ascending tracts. So in any ascending tract, we have 3 neurons in the pathway. So therefore we have 2 synapses. One that happens somewhere within the spinal cord or brainstem, and then one that always happens in the thalamus. So one of the synapses that happen in any of the sensory pathways that I'm going to mention now, one of the synapses will happen in the thalamus. The other synapse can happen anywhere else, and we'll discuss where those happen, but there will always be a synapse in the thalamus, because the thalamus is essentially the sensory relay point. It modulates all sensory information that is coming into the brain and then processes that and then sends it out to the appropriate area of your primary sensory cortex. Okay, so the first sensory pathway I'm going to mention is the dorsal column medial lemniscus tract. So the function of this tract, or the sensations that are carried within this tract that you then become aware of, are things like discriminative touch. So for example, I'm holding a pen right now, and even with my eyes closed, I can move the pen around my hand and I can feel that, there's a clicky bit, there's a sharper point at the end where the tip is, and I can identify that is a pen. If you had a ball in your hand, you'd be able to identify a ball or a bottle, whatever. So that is discriminative touch. Also proprioception, so that is the conscious awareness of your body's position. So for example, I'm sat at a desk right now, and I am aware that I am sat on a chair, my knees are bent, or flexed, my hips are flexed, my spine is straight or relatively straight, depending on what my posture is like at any particular point. So having an idea of where you are in space is crucial, so essential when you are planning a movement. And so it's essentially like the starting point. And also vibration sense is also picked up via the dorsal column medial lemniscus tract. So, as I said, we have 3 neurons in the pathway. The first order neuron is a peripheral nerve. So it could be, well, let's use T1 again, since we used it in the last example. So let's say I'm holding my pen. I have my pen in my hand, and I am aware that that pen is in my hand, or I'm moving it around and I know that there is a pen in my hand. So that sensation is first picked up by receptors, touch receptors within the skin of my fingers, which are attached to the first order neuron in the dorsal column medium lemniscus pathway. So T1 nerve is going to come from the periphery. It's going to run through my hand via the spinal nerve. It's going to run via my forearm and arm back to my spinal cord. So let's use my left hand as an example again. So so the first order neuron is going to enter into the left side of my spinal cord at the level of T1. It is then going to run up in the dorsal column, so at the very posterior part of the spinal cord. It's going to run up on the ipsilateral side, so it's going to stay on the left hand side until it reaches the medulla. And within the medulla there are 2 nuclei that correspond to this pathway. There's a nucleus gracilis, where any neurons that have come from the lower limb are going to synapse, and there is a nucleus cuneatus, where neurons from the upper limb are going to synapse. And because this is coming from our upper limb, from our fingers via the T1 spinal nerve, our first order neuron is going to synapse in the nucleus cuneatus. After this synapse has happened, our second order neuron arises from this synapse and crosses at the level of the medulla. So almost immediately after the synapse has happened, the second order neuron decussates onto the contralateral side from where the signal came. Okay, so now the information is traveling on the opposite side of the body. So it's going to go to the left cerebral hemisphere. So it travels up through what we call the medial lemniscus within the brainstem. It enters in to the cerebrum via that cerebral peduncle that I mentioned earlier, and it's going to go into the ventral posterior nucleus of the thalamus. And at that point, the second order neuron is going to synapse with the third order neuron, because what did I say? All sensory information all sensory information that is traveling in a sensory pathway is going to synapse within the thalamus. So here we have that synapse. So the second order neuron synapses in the VPL nucleus of the thalamus, and the third order neuron exits the thalamus, enters into the posterior limb of the internal capsule, up via the corona radiata, and is going to go to the hand area on the posterior post-central gyrus, or the primary sensory cortex. So that is the dorsal column medial lemniscus pathway or tract. And the final pathway that I'm going to mention is the spinothalamic tract. This carries information or sensations of pain, of temperature, of crude touch being sort of, so if you put your hand down on the table in front of you, you're aware that there is something there. That is crude touch. Or if you walked into a glass door or something like that, you would be aware that there is something there. That is crude touch. And also pressure. So just like in the dorsal column medial lemniscus pathway, we have 3 neurons in the pathway, therefore we have 2 synapses. One is going to be elsewhere within the pathway, but one of them is definitely going to be within the thalamus. Okay, so let's say that you have just pricked your right index finger on a pin. There's going to be some pain, okay? You're going to feel it. So you're going to have pain receptors from 3 nerve endings in your skin. These are connected to neurons that are going to run with it as part of spinal nerves from your right index finger to your spinal cord. And the level of the spinal cord that it's going to go back to, well, let's just use T1 again. Okay, so I've swapped over sides now, haven't I think? Yes, compared to the last one. So we're going to end up on the right side of our cerebral cortex. Hang on a minute. So we're going to end up on the left side of our cerebral cortex. Yes, so it's our right finger that we have pricked. So the sensation travels via the T1 spinal nerve and enters via the dorsal horn into the spinal cord. Okay, and in the spinothalamic tract, the first synapse happens at the spinal level where the spinal nerve has entered into the dorsal horn of Gray within that spinal cord. So that is where our first synapse takes place. So the second order neuron then immediately decussates. It decussates via the anterior white commissure, which is this little bridge here, and then it runs into the contralateral from where the signal came to. So we're now on the left side of the spinal cord. It enters into the spinothalamic tract on the contralateral side of the spinal cord. It runs up via the spinothalamic tract all the way through the spinal cord and then into the brainstem, where the spinothalamic tract just becomes the spinal lemniscus. It runs up through the cerebral peduncle into the thalamus. What's going to happen in the thalamus? There's going to be a synapse. So the second order neuron synapses with the third order neuron. The third order neuron then enters back into the posterior limb of the internal capsule and then runs up to the hand area of your cerebral cortex. And that is how you would perceive that pain in the tip of your finger. Or if you pick up a cold can of something from the fridge, that temperature change, the awareness of the temperature of that can or whatever it is travels via the spinothalamic tract to make you consciously aware of a temperature or a pain of something you're touching or pressure. And that's it. Thanks very much.